Thank you for the invitation to come here today. You need to hear me, so I'll take my mask off. As I explained to a person on the plane who's a bit skeptical about wearing this, if that person had been in as many intensive care units in as many countries in the last few weeks as I've been, or been on call on Monday night and in clinic on Tuesday, who knows what weird stuff I've been exposed to. So I'm doing it for you. But I think the trade-off now is that communication is what we're talking about, and trade-offs in terms of risk is going to be a germane thing, judging by the zest in the discussion this morning. So I have some disclosures. They don't much obtain here. I've done a lot of work on frailty and have gotten several scales and stuff that we use for that. And I've asserted copyright on them, because if you don't do that, other people try and sell your work. So if you ever want to do stuff on frailty, and you might well want to, don't buy it, just get it. It's all free online. So here's what we want to talk about today. Sorry. I see. I'm pushing buttons. I can see them here.
Sorry, that's another tech issue. Sorry.
Fine. Okay. So you'll just have to listen to me. So there's a couple of things that I want to do. One is to outline the rationale. So I don't have data to present today. I've got a protocol, and the reason for the protocol is to get at one of the issues we've just heard about is an underpowered study. So what we're aiming to do is to share this protocol, and that way other people can do things that might align with that. And as a consequence, there's a whole bunch of other things on the screen. I'm not sure I'm supposed to press any of them now. I will try. No, it's made everything go away. Anyway, so the idea is that we're going to try and share the information, share this protocol, and other people who might want to do a similar type of work could have outcomes that would be similar to what we've done, plus, and then we could see whether power would help if we've got a negative study. Other than that, one of the things you've learned at my age, or I've learned anyway, is that medicine is a humbling business, and we may well be repeating at least one of the problems that you have talked about. So we're starting with a cluster randomized, which is how the studies have been done so far, because you can't do the randomization at the level of an individual. Placebo-controlled trial of Far-UVC light to reduce the incidence of respiratory viral illnesses in long-term care. And I want to point a few of the novel things that we have done, including some of the measures we have introduced to better detect these infections in a nursing home environment. So the dilemma in a nursing home environment is that the usual triad of wearing a mask, washing your hands, and social isolation is not pragmatic there, because there's a high incidence of people with physical frailty or with dementia, which impairs their ability to adhere. And so because of that, we are very keen to find other things that we might do that would add to the traditional measures. Now, I can tell you that we had some dramatic evidence of benefit, both from masks and from infection, and from vaccination in this study, in as much as very quickly after the first two waves, the incidence of COVID-19 went way down. And for 2 years, for the first time in living memory, we had no influenza cases. So it's clear that it's the combination that winds up being important, but to have no cases of the flu is quite a remarkable thing for a nursing home. Also in the nursing home, personal care is in close proximity, and there's no getting around that. And the people who are in the long-term care facilities themselves, they don't get, they're not where this is introduced. It's coming in from the outside. And the other thing we found, which is very striking, is that physical distancing actually makes the outcomes or the lives of the residents worse. And so because of that, again, we were keen when we heard about Far-UVC light be involved with that. Okay, now my button isn't working either. Okay, so we started the trial in 2021. We've just introduced a second phase of the trial here now. I see things happening down here, which give me hope. Technology, I tell you, you got to be careful. So let me just go through these other things here now. Okay. So we've updated the lamps. We've gotten ones now that can give us a higher dose. How much higher, you ask? It's within the new ACGIH threshold limit value as measured per ANSI RP 27 standard. We also have filtered lamps and diffused light done by certain engineering calculations for beam angles and such. The first study that we did was for 12 months. We're going to, in phase 2, extend that for 24 months. So we'll have 36 months in total. So again, expand the sample size a fair amount. The ambient mortality rate in long-term care, it used to be about a half for years. It's now about a third. So it does, with the passage of time, mean we will be exposing more people. I'm just changing this because I need to see what my notes are. Let me just make sure I haven't forgotten anything thus far. No, and we're going to add a third site, potentially a fourth. So here's a thing to pay attention to if you're going to do this sort of work. Here's how we started. Its technology is a vulnerable population. The people who love and care for and the family members of this vulnerable population, they are very concerned about what goes on in long-term care. And so we made sure that the first thing that we did was a qualitative study in which we went and talked to the administrators, the frontline staff, the families, family councils, the resident council, with a fairly small number of the residents who can speak for themselves. And we listened to what everybody had to say after we described to them what it was we were trying to do. And we listened to their concerns. We paid attention to what they were seeing online. We gave educational talks, but we had people, not me, we had women for the most part who went around and did qualitative studies. And that was work in progress, I guess. Although it's gone here now. And that was a key aspect to what we did. And we also had a commitment that we would pay attention to, sorry, we also said we pay attention to what their concerns were and try and address them. And that wound up being very useful in a bunch of ways. One of which was, to be honest, we didn't get any pushback from people. So we started by going to an old downtown nursing home, public sector, not-for-profit, loving place, but from the layout standpoint, the low ceilings and windows that open and those sorts of things. And we went in there after they had a terrible experience. So 1 out of every 7 people there died in 4 months. So against that background, when the CEO said, I want to put hand on heart and say, we have done everything we could do, that was really this, we heard variants of that everywhere we went. You know, we did have folks who looked stuff up online, had weird sort of theories and whatnot, you may well imagine. But as a general proposition, everyone signed up fairly quickly. Okay, all righty. Well, someone else's slideshow is up there now. So I'll tell you that the next thing that's key.
Kenneth, I wonder if we should not take a break. This is not fair to you, and let's take a few minutes break. Try and get this thing working. We'll come back in 10 minutes or something like that.
Actually, so I'm glad for that. But the thing that I want to tell people about next doesn't really require the slides, and it's not engineering. But I think it's really important for whomever might want to undertake a study like this. So I'm happy to press on with this part of it. Okay.
Okay. Well, you're very brave, and thank you.
I used to be a fireman. Brave is a very relative term. Fantastic. Okay, blah, 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 blah. Okay, so what we learned from phase one, the qualitative study, stakeholder engagement, all that sort of stuff. We're still blinded to whether what happened in phase one worked, because we realize that we're on the underpowered side. But the best thing is to go for it. And if we unblind ourselves, then we've wiped out information. We've wiped out the strength of what we have otherwise. So we are going to look at efficacy and safety, and what we're learning in the last couple of days is the safety bit is more complicated. We had actually gotten that in front of this a bit, but pay attention if you think that you can help us with the safety stuff. I'll be talking about that in a bit. So the primary objective was to see whether or not we could reduce the incidence of respiratory viral infection, so more than just COVID-19, and we're going to do case definition by a positive swab. And then we've expanded it to look for SARS, influenza A, influenza B, respiratory syncytial virus, and others. And we've had some experience with all of that, including the 254. I know physics of it doesn't work, but it was extremely important for the population that we said. We will look at that too, just in case. And then one of the things that we have done is the lamps are only available in the common areas. I'll show you some floor plans next. And part of the reason we did that was cost, but this would have to be paid for by a provincial government, and it wouldn't be cost effective to do it in residence rooms. I think we stumbled onto a great way to be able to limit the dose as well. So to the extent that that works in our favor from safety standpoint, that's a good thing. And then we're going to look at some screening tools. Is what I want to talk to you about next. And what we've added in most recently for phase two is to check the ozone emissions of that. And I'll tell you a little bit about that too. So it was the realization that the power is low here, that the easiest remedy for this is just to stay in the sites that we're at a site, but then extend the exposure time, because that means with the turnover in the population, the sample size will go up, and we get to something that would approximate the type of impact worth paying for. And that was the other part, that because this is very much being done to see whether it would help the way we provide care in our province, which is Nova Scotia, about 1 million people. Beautiful place to go, by the way. That was the how we tried to make virtue of necessity. So site one, 95 study participants, 2 clusters, 1 active, 1 placebo. So this is in a modern long-term care facility designed with all the right groovy stuff. There's enough room, and there's enough space for people there. They're virtually all single rooms. There's common dining areas. There's common areas for recreation, but they're common to what is the doctor and hospital called the ward. There's called the neighborhood. And so we are able to have groups that pretty much don't mix. So the people who care for them do. And the other one was Northwood. which was the old place that I had described to you. So, and there's a third site that comes in when it kind of seems to mix aspects of both, but an important difference here is that the common area will receive some residents from each place. So there's a potential for mixing there. So the sites are variable, which from generalizability bit is a strength, which includes the number of lamps that are used. But the general idea is that everyone has an equal opportunity to be in the common areas in their neighborhood. And it's the common areas that the calculations and the dosing have been based on. They're installed in high traffic areas. Cycling is leftover from the first phase. They're on the entire time now, thanks to the new technology that we've got. And we've also got placebo controls. So I told you about how we started things off. And I just want to go over a little bit the ethics of it, because I realize I'm speaking to Americans for whom rights are foundation. You can't imagine anyone would think otherwise, and yet here we are. We're not really a rights-based culture in Canada, which growing up in I think is a good thing. But the pragmatics of it was that no individual resident has the ability to withdraw from the study. They'd have to move their rooms and those sorts of things. And for the most part, once people have settled into an environment, there's no real pragmatic preference there. So if anyone really wanted to, or the families wanted to, they could, but generally no one has taken us up on that. So we got an ethics waiver which said what we need the informed consent for the individuals or their participants was to use their data, but we didn't need informed consent for them. Like they couldn't object to this happening on the floor that they're on. And in the end, no one chose to move from that. And I made the point about how important the stakeholder engagement was, and we really put money and time behind that. And what we were keen to do was to make sure that the research was as unobtrusive as possible by taking advantage of measures that would collect the information that was going to happen anyway. So it was the case, the mandate for the nursing homes was that every day, every patient had to be screened using public health measures. So the thing I was going to keep on and tell you about, I'll tell you about now. I'm a geriatrician. One of the things that we know, swear the oath, is that disease in older people who have many things wrong does not present in the same way that it will in people who've got just one thing wrong at a time. This, in fact, is a huge challenge for society, but I won't talk about that now. Just to say that, for example, an older person with pneumonia, full-blown, honest to God, that they may not have a cough, or they may always have a cough and it hasn't changed much. They commonly will not have their temperature go up. In fact, it can go down. It's a more specific sign. They might or might not be short of breath, or if they are, they might or might not complain of it in the traditional way that we expect. So we can't use only the public health guidelines. But what I also know as a geriatrician is that when frail older adults get ill, typically one of three things happen. Either they get confused, and if they're confused, it gets worse, or their mobility gets worse, or their functioning gets worse. Now, the pragmatic aspects say, okay, well, that's fine, but you've got people who, an important fraction are bedfast. How are you going to look at the mobility in them? Well, it turns out one of the instruments that we had worked out from long ago looks at how older people move in bed when they are ill. And we're able to quantify that, and therefore we can screen, and if it's positive, do an assessment. And the same way for confusion, there's a thing around called the SQUID, the single question in delirium. And so if that's positive, then we do the delirium assessment. And the thing about function, which is really interesting, there are some people who they are so impaired that they have to be fed. Now in Canada, you never see an NG tube down someone's nose. in long-term care. Everybody's hand-fed. But if you hand-feed people, one of the things that you understand is that when you introduce the bolus of food to their mouth will open. If they don't, that's the equivalent sign of confusion. And for the most part, when they get it on the front part of their tongue, reflexively they'll put it to the back of the tongue from which they will swallow. But many people who are impaired, they'll come to the point in which they put it on the front of the tongue, and instead of it going to the back, it gets over to the side and gets caught in here between the tooth, the teeth, or the gums, and the inside of the cheek. That's called pocketing. So as we spoke with the frontline care staff and talked about the issues of how it is we would detect impaired function, this was what they hit on, in addition to all the other things. But for this special case, that's what they hit on. And that's what we learned from that, from talking to them. When we say we went and talked to people, it wasn't, here, sign this, be okay. It's what do you have that can contribute? And I think that's been a real part of the study. And if another group is going to do it, I would really strongly suggest to integrate that. That also makes the time to or the commitment that you need from people to show that you're willing to act on your commitment to them. So we had a number of things to we put a lot of effort in being transparent, and a lot of effort in sharing information, asking questions, and giving people the chance for feedback. I think that is really time well spent. When we went to the new sites, we started from scratch. At the old site, we just said we have an update now with this fancy new technology. We have ongoing anonymous surveys that are available. We give feedback as problems come up, and then we paid some attention to what the nature of being a research participant is, and we've aimed to honor that by collecting data that's useful and not obtrusive for them, and then also by keeping everything private. This, we've talked about the alteration of consent. We've talked about this. There's a control arm with the sham devices that are there. We're building on top of everything else that we do. This is not the right picture. This is the 1.0. We've got the 1.5s now. This is how we did the placebo component of it, and we've been vigorous about maintaining the blinding. So there's only one person who is an experienced clinical trials coordinator, and she has a poker face as a natural thing. So she walks in and points up the wee device meter thing and finds out whether there's radiation or whatever the right word is coming out of it, and writes it down sternly and goes to the next one and such. So we routinely monitor what the output is and do it in a way that make sure that it's not blinding. These are the things are clean. We tried our best to make sure that we've got a worthwhile intervention. Now, the ozone testing. So we're learning a lot about this. And just last week, we met with people from the National Research Council in Canada who kindly gave us access to a fancy 0.01 parts per billion meter that we could use to measure ozone. And the initial results of that looked to be quite safe, but we know we need to be more vigorous about that. Now we can't just do an occasional one. So that's the thing I'm very open to. If this is within your technical expertise, we'd be keen to talk to someone about that. So the trial timeline, so the outbreak started in actually April to May was when it really hit. It took till April, May 2020. It took till November 2021 by the time we got the lamps and the ethics and the people and the money and all that sort of stuff that needs to be done. And June 2023 is right now when we started phase 2. And we're going to wait till we get to the end of the data collection period for statistical analysis. There is a blinded interim analysis in which we just look for differences without knowing what the group assignments are, and ultimately we'll have a cost consequence analysis to come with that. And we're doing these other very straightforward sorts of ways to describe what it is that we're finding. We're tracking person time in this study, so it's all going to be about infection by rates, taking into account the amount of time people are there. Up till now, we've had 91 positive cases of COVID, 7 of influenza A, and 4 of RSV. We've had some evidence of co-infection. No one has died with any of these illnesses. Again, quite a remarkable fact. So, there are these limitations that we're aware of. So we talked about it has to be plus or randomized. We've talked about how the consent issue has gone. We are interested to find out how often infection occurs, and then we're interested to find out what the differences between the groups are, and we're only doing it in the common areas. Is it better or worse to get in to their bedrooms or not? We're not sure. I'm thinking by what I've heard right now, it may be too soon to go with 24-hour coverage. And we're at the funding limit for the sample size, but we're hoping that if we work with making the protocol transparent, that would be helpful for subsequent meta-analyses. So what's novel about this, the technology is an integrated study design looking at multiple outcomes. Is we have been, we didn't in the first round, but it's a condition funding for the second that we're doing the wastewater analysis by another group along the side of that. We've got various experimental methods, controlled trial, qualitative methods, the economic analysis. And one of the other things that has been very, very useful to us is we engaged the leading people in infectious diseases and medical microbiology to come with policy analysts and people from the Department of Health, and so we've got a good interdisciplinary group who is there to give us advice as we go forward, and we're keen to participate in environments like this and see what other information that we can get and how much more we can collaborate there. And we've made some innovations to the protocol, which we think will enhance the quality of research in long-term care as a general proposition. So I've got a number of people to acknowledge here. The funding is from Research Nova Scotia, which has a very impressive new approach, which is a co-funding model and a co-design model for projects where there would be a benefit to the province itself, and then a large number of other groups there. So I thank all of them for their help, and I thank you for listening. Thank you very much. I have left time for questions.
Yes, okay.
And comment.
There's a comment right here in the front row. And we'll go to Ed.
Thank you. First of all, congrats on an awesome talk, given all the technical difficulties. It was really very engaging. I have a couple of questions. In a standard type of clinical trial, like for drug development, sometimes there are interim analysis to find out, is the active really worth breaking the blind in order to save more people? So that's one question. And the second is, are you collecting all the data about other medical issues, like how many are vaccinated? Did they get their boost? to what diseases, et cetera, so that when you get the data, you can analyze if there's some demographics there?
Yeah, the answer to the first question is, yes, we have done an interim analysis and still safe to proceed. The second thing, I sort of spared you all the stuff that I would do for an audience of my geriatrician colleagues, but we've done a comprehensive geriatric assessment on everybody at baseline. We sort of know everything about them, and we have put effort into being able to measure their cognition, mobility, and function at the start, and then we'll see how that plays out. And then everybody's on screen each day, which is giving us a boatload of really interesting data.
I look forward to the publication.
Yeah, I remember your presentation online, and I was envious of the first building that you showed because it had these two very separate wings. I had inherited a school study where we, which had soft outcomes, and so introduced the idea of wastewater testing, which I think you just mentioned.
Yep.
And we have a poster, P26 upstairs. It was only a pilot study, 4 months, but I do think that the pandemic has given us a powerful tool to look at building-related airborne infection, and not just COVID, but influenza, a variety of things you can measure in wastewater. So I would just throw that out as for the general audience here, that I think, particularly in long-term care, in schools it was more difficult because kids can be infected at home. But in long-term care, it's really probably going to turn out to be an optimal way to look at this.
Yeah, so we certainly have our fingers crossed on that. We had tried to get it going partway through the first phase, and it turns out that in Northwood, which is old, and the water piping and whatnot is old, they actually had problems to locate exactly where the exit was. Like, this is non-trivial undertaking. So we're very keen to get all of that working.
There was another kind of way.
Hi, I'm Michael from Frida Tech in UK. Thank you for getting through that. A question which you might not be able to answer to is whether or not you've noticed a reduction anecdotally through comorbidities like MRSA, C. diff infections through this cohort of other illnesses you'd expect to have in this nursing home population, but have not seen as much of during this period.
So again, we've been very careful about that because that would get us to unblinding. So that information is available, but it has not been shared in any way, and I don't know it, for example. So.
And first, let me just say that how much I admire and appreciate all you're doing for those residents in your community, so often neglected. It's absolutely incredible. And as for the question, with your real use case scenario, when we talk about practical applicability, what are we actually seeing out there? And also in terms of the risk models that we're looking at, what is the volume of early signal that you've been seeing, or not seeing for that matter, of early indicators of VOC and ozone toxicity showing up in your residents?
So we would be unable to distinguish anything specific for that, right? So what we have been looking at is change in health, and then that would get the usual workup that would be done for that. So respiratory illness in older people is common. Much of it is non-infectious, and if you were to include heart failure in it as part of that, lots of people have shortness of breath, for example, or the functional equivalent for that. So I'm not able to comment on anything specifically with regard to that, and I don't think that. So I imagine it would be fairly difficult if you were to work through the differential diagnosis of why someone is short of breath to get to a VOC component in a very frail population. My brother is an orthopedic trauma surgeon, and he sort of has a qualitative scale that he would describe risk in a group like this. And it's based on procedures that people have a pretty good understanding of what the level of risk would be. And most of the people there, I think if I was going to use his scale, he would call them high risk for a haircut. So this is a very high risk group.
All right, maybe one more question. All right, thank you, Dr. Rockwood.
Thank you.